Okay, good morning, everyone. I'd like to welcome you to this meeting of the California Technology Assessment Forum of ICER, the Institute for Clinical and Economic Review. Thank you. Today we'll be discussing oral immunotherapy and Viaskin Peanut for peanut allergy effectiveness and value. Before we get started, we'll begin with introductions, and it is important that we all acknowledge any conflicts of interest. Say every panel member here on CTAF has already met that conflicts of interest policy, but as you introduce yourselves, please let us know if there's any update to that. We'll go in clockwise order and then have our experts then also, when you introduce yourself, give us any potential conflicts of interest. Again, I'm Paul Heidenreich, chair of CTAF. I'm a clinical cardiologist at Palo Alto VA and Stanford University.
Rena Fox. I'm a professor of medicine in the General Internal Medicine Division at UCSF.
My name is Sei Lee in the Division of Geriatrics at UCSF and the San Francisco VA.
And we'll say, and no updates.
Oh, no updates for
No updates.
Felicia Cohn, Bioethics Director for Kaiser Permanente Orange County and Clinical Professor at UC Irvine in the Department of Medicine. No updates.
Ralph Brindis, I'm a clinical professor of medicine at UCSF in the Phil Lee Institute for Health Policy Studies, and I have no updates.
Kimberly Gregory, I'm a Vice Chair of Women's Performance and Quality Improvement at Cedars-Sinai Medical Center. I'm a OBGYN, maternal fetal medicine specialist, and a professor at UCLA, and I have no updates.
Hi, I'm Jeffrey Hotch. I'm professor and chief of the Division of Health Policy and Management in the Department of Public Health Sciences at University of California at Davis, and I'm also the associate director for the Center for Healthcare Policy and Research. I have no updates.
Good morning, Neil Kohatz. I'm an independent consultant in population health and no updates.
Lisa Murphy. I'm a clinical professor of clinical medicine at UCSF and an endocrinologist based at San Francisco General Hospital.
I'm William Weemack. I'm a patient advocate, and I have no updates.
I think it's on, yeah. I'm Joy Melnico. I'm a professor of family and community medicine at UC Davis and director of the Center for Healthcare Policy and Research at UC Davis, and I have no updates.
Hi, Alex Smith, associate professor of medicine. I'm a palliative care physician at UCSF and the Division of Geriatrics, and I have no updates.
Robert Rentschler, retired oncologist, no updates.
Jeff Klingman, I'm the chair of neurology at Kaiser Permanente, Northern California, no updates.
Richard Seiden, patient advocate, no updates.
And I'm Bob Collier. I've been a patient advocate for going on over 28 years now, specializing in research. I have no conflicts at all.
And now we'll ask our experts.
Hi, I'm Nuri Hong. I'm Chief of Strategy and Innovation at FAIR, or Food Allergy Research and Education, which is a national advocacy organization focused on food allergy. Our conflicts are disclosed in the report, but I'll read them for full clarification. FAIR is a 501(c)(3) public charity and receives financial support from multiple sources. In the past, FAIR has received general support grants for corporate sponsorship from each of DBV and AImmune. These general support grants are charitable in nature and do not require FAIR to perform any service on behalf of DBV or AImmune. At one time, FAIR was a shareholder of AImmune, but those shares were sold in their entirety in 2016.
I'm Caroline Moassisi. I'm a patient advocate and a parent of 2 children with life-threatening food allergies.
I'm Ruchi Gupta. I am a professor of pediatrics at Northwestern and Lurie Children's in Chicago, and I'm director of the Center for Food Allergy and Asthma Research there.
Matt Greenhot. I'm associate professor of pediatrics and director of the Food Challenge and Research Unit in the section of allergy at Children's Hospital, Colorado.
Okay. All right. At this point, I think we'll go ahead and I'll ask Dr. Steve Pearson, President of ICER, to give us an introduction.
Good morning, everybody. Thanks for being here. If you would, please turn your cell phones to stun so that we can avoid those events. Welcome and thanks for being here. For many of you who have helped us throughout the process of developing this report, the public meeting serves as a very important milestone. So why are we here today? One of the chief reasons is that we often meet around new interventions in health that address a significant unmet need. Here I have a quote, and we talked to many different parents and parent groups. One said, "My son struggles daily at school. He does not feel safe when I'm not around because he's worried that no one else will know what to do if he has a reaction." This is one of those classic conditions and set of interventions in which the family is a very important player, not just the patient. So for caregivers, here's a quote that says, "I feel I operate under higher levels of anxiety all the time, particularly when my son is out of my care. He's never had a severe reaction, but one day I got a call from his school that they suspected he was reacting to an unknown exposure, and I had to rush over to his school to assess him myself. I have frequent flashbacks to this and imagine it happening again." This is from a parent of a child with a peanut allergy. So in addition to the unmet need and the impact on the family and other caregivers, we are talking about new treatments that have mechanisms of action, and anytime we have new treatments, they raise questions about the appropriate use. Which patients can benefit most? What about its cost? Who bears that cost and where in the system? And we also know that this occurs in a context in which increasing healthcare budgets are increasing expenses are really straining healthcare budgets at all levels. It affects individuals, state and federal budgets, all the way through. So we think that it's a really important function to have public meetings looking at independent assessments of the evidence and to try to work our way towards a better understanding of how to bring new interventions into the care system in a way that maximizes their value, maximizes the ability of patients who can benefit to reach these new treatments, and do so in a way that's ultimately sustainable and affordable, both for patients and for the healthcare system. So you've been introduced to the members of the California Technology Assessment Forum. They are here to think hard and to discuss in a public setting all of the issues around evidence and other things, because there are always other aspects that we need to consider when we're talking about value in the broader picture. We at ICER are, in a sense, the academic secretariat, and we convene these meetings. So just briefly about ICER, we're an independent, not-for-profit research organization. Our funding, which we keep updated on our website, is shown here in this pie chart, where you can see that approximately 77% of our overall revenue comes from a set of nonprofit foundations, chief among which is the Laura and John Arnold Foundation. There are other funders, including the California Healthcare Foundation and others. We have a separate policy summit program where we do take direct contributions from drug manufacturers and from health insurers and pharmacy benefit managers to run a program for them where we convene them and talk about evidence policy, create white papers, but that money is not used to support the research efforts on our reviews. So the report today that serves in a sense as the academic underpinning occurs in development over a long period of time, almost 8 full months. And it begins with a really critical phase that we call scoping. That's the phase in which we go out to the manufacturers, to the patient groups, to clinical experts, and we say, tell us about this condition. Tell us what it's like to live with this condition or to be part of a family in which you're grappling with this condition. Tell us about the diversity. Tell us about what really matters to patients and their families. What does value look like to them? And we use that as our North Star, if you will, when we start off looking for evidence and bringing together the different pieces that will ultimately comprise the report and this meeting. So we do an internal ICER staff review of the available evidence. That might be the published evidence, and it might be new survey data. For instance, we'll hear some of that today. We also have a cost-effectiveness model that's developed by academic colleagues. Today, they are from the University of Washington, and you'll be introduced to them soon. We go through several iterations of public comment and revision, and the report has also been reviewed by experts, including Dr. Gupta and, I'm sorry, not Dr. Hong, just Mr. Hong? Mr. Hong. And we consider experts from the patient community to be as important as clinical experts in all ways. So how is the evidence in the report structured to in a sense, underpin the discussion today? We think it's important to note that we do have a goal. It is sustainable access to high-value care for all patients. Sometimes we even condense that down to fair price, fair access, and future innovation. To get there, we believe that it's important for policymakers to think about two major conceptual buckets of information. One is around how we judge the long-term value for money of new interventions. And the other is the short-term affordability, because sometimes they can be in conflict. We can have good long-term value, but in the short term, there's going to be a big budget impact that needs to be managed. So long-term value for money itself is a complex set of considerations. And we start with a clear-eyed look at the evidence on comparative clinical effectiveness. And that, again, can include evidence that hasn't been in the public domain before. We're trying to either generate new evidence or make sure that we're aware of the evidence that can make the best decisions possible at this point in time. We develop an incremental cost-effectiveness model, and then we'll be talking a fair amount about two other important elements. One we label as potential other benefits or disadvantages. Those are aspects of value that may be very important to patients and their families or others, but that aren't really fully captured in the clinical data or in the cost-effectiveness model. Similarly, contextual considerations try to make sure that we are taking a broad look at the ethical and the social values that may pertain to this particular area. So wrapping that up in our report is what we try to do in a way that will support today's discussion, but you'll hear a lot about each of these different buckets. The short-term affordability really considerations are supported by a potential budget impact analysis in which we just try to create a graph so that people can do their own estimation of at a certain price and at a certain use within the eligible patients, what is the short-term budget impact over the first 5 years that the health system is going to want to address. So that's the backdrop. The agenda for today starts with a presentation of the evidence. After I'm done, you'll hear from Dr. Jeff Tice, and then a caregiver survey presentation from Ken Mendez of the Asthma and Allergy Foundation of America. And then we'll have the cost-effectiveness results summarized by Greg Guzauskas from the University of Washington. We will then have a phase in which the manufacturers will be able to provide public comments, and we'll have discussion around their comments and a public comment further after that. Lunch at noon will be provided for visitors out here in the hallway. The CTAF panel itself will eat in the separate room, but we'll have lunch available for attendees. Then we'll come back to do further deliberation on the evidence with a vote from the CTAF panel on different aspects of the evidence on both clinical effectiveness and potential other benefits and contextual issues. We'll have a short break, and then we'll do a policy roundtable. And I'll tell you now, just a teaser for what that is. After having discussed the evidence and had testimony and wrestled with it, we want to turn our eyes forward and talk about what's the best way to translate that evidence into a future of coverage, of pricing, of education, of future research. What's the best way to use where we are today to build a system that's going to work best for patients in the future? And work best is going to be from different perspectives. It's going to be from the perspectives of patient representatives, clinical experts. We'll have manufacturers, and we will have a payer represented on that roundtable. So then we'll close with brief reflections from the CTAF panel itself, and we'll be adjourned by 4 o'clock. So with that, let me just reintroduce our clinical experts. Since they were nice enough to read their conflicts, it's up here on the board, along with their distinguished academic and clinical pedigree, if you will, and our patient experts, again, for whom we have a lot of thanks for their participation. All right, with that, let me introduce Dr. Jeff Tice, Professor of Medicine at the University of California, San Francisco, who will kick off our evidence review. Jeff?
Great. Thanks, Steve, and thanks to everybody for coming and participating in the discussion today. I look forward to hearing from many of you. I wanted to start off by acknowledging members of the team that helped develop the clinical evidence review, Serena Heron-Smith at ICER and Judith Walsh at UCSF. And again, we have no conflicts of interest relevant to this report. So as most people in the room are aware, peanut allergy is the most common food allergy, childhood food allergy. Dr. Gupta helped to develop the most recent epidemiology on this. There's still some controversy, but around 2% of children have a significant peanut allergy. Reactions are common, but death is rare. So accidental exposures occur in 5 to 20% of patients annually. Anaphylaxis is less common. Estimates range from about 1 to 2% per year. Those you'll see in the clinical trials, the rates were actually higher in patients in the clinical trials. But mortality is uncommon. So one estimate looked over at mortality due to peanut allergy over a 10-year period, and the average was about 4 deaths a year. So mortality is minimal, but still a driver of fear for patients, especially for parents or caregivers of children living with this allergy. We've already heard from Steve, and we'll hear more from the AAFA about the impacts of of peanut allergy on social activities, parental anxiety, quality of life for both children living with one of these allergies and their caregivers. And approximately 15 to 20% of children with food allergies will outgrow them by adulthood. Current treatment, really there are no treatments other than avoiding peanuts and immediate use of epinephrine in the case of an exposure with a reaction. There are no FDA-approved treatments yet, though we're considering 2 therapies that have been submitted to the FDA, or one will be resubmitted soon. The goals of the new therapy are to reduce reactions to accidental exposure. This is really what desensitization is all about. It is not a cure. And the hope is that the desensitization will lead to improved quality of life, reduce anxiety. But really, the core of current treatment really has to remain in place. So everyone needs to realize with these new therapies, children still need to avoid peanuts strictly and carry epinephrine in the case of an exposure leading to a reaction. So the primary therapies we're considering are AR101. It's a standardized peanut flour that's taken orally, started at a low dose, and every 2 weeks, if tolerated, the dose is increased to a goal maintenance therapy of 300 milligrams of peanut protein daily. Each dose escalation requires a visit to a physician's office for monitoring with the higher dose, so a significant burden for the children and their caregivers. And that's a minimum of 12 visits because sometimes there are problems escalating. People go up slower, go back down to another level, but a minimum of 12 visits. Viaskin Peanut is a different technology. It's a transcutaneous approach. Patients wear a patch in 6 different locations on their back. They're rotated. The number of hours they wear the patch starts at 6 hours, and it's escalated over 3 weeks to 24 hours a day. Delivers 250 micrograms, so less than 1/100th of the dose of AR101, but transcutaneously. This technology does not require additional visits after the first one when increasing the amount of time the child wears the patch. For oral immunotherapy, there's really no standard. There are many different treatment regimens that we'll talk about briefly. So in our systematic review, we found two randomized trials of AR101, the phase 3 trial, the Palisade trial, which we'll talk about in detail today, and a phase 2 trial published in 2017. I will say, and I'll talk about this briefly, there was a European meeting the first week of June in which a second phase 3 trial of AR101 was the results were presented, and I'll go over those briefly at the end of my slide set. Viaskin Peanut, one phase 3 trial, the Therapies trial, and two phase 2 trials, all of which have been published now. For oral immunotherapy, only 2 trials met our inclusion criteria. There are a number of other trials, and I'll talk about a systematic review that was published recently. Experts encouraged us also to present some case series with large numbers, and those are included in the report, though I'm not going to talk about them today. We elected, sometimes when we have a number of different therapies for one similar indication, one disease state, even if there aren't head-to-head trials, we'll do indirect comparisons using a statistical approach called network meta-analysis. In this case, we felt like the patient populations were sufficiently different, the outcomes in the trials were sufficiently different where we didn't feel it was appropriate to do a network meta-analysis. In particular, one therapy, the Viaskin Peanut, has primarily been studied in children ages 4 to 11, where AR101 has been studied primarily in a slightly older population, starting at 4 as well, but up to age 17. They also, in the phase 3 trials, had different entry criteria, in particular, a maximum tolerated dose of 100 milligrams of peanut protein in one and 300 milligrams in the other. And also the outcomes were very different, and I'll go over those in a little bit more detail, but they're sort of standard dose escalation protocols, and they varied in the 2 studies. There were different definitions for eliciting and tolerated dose, and the criteria for stopping the dose escalation in the oral food challenge were slightly different, making it challenging to compare the results head-to-head. So for oral immunotherapy, there are 2 poor quality randomized trials and 2 case series. All of them started at low doses and increased to a target maintenance dose. The maintenance dose ranged from a low of 125 milligrams of peanut protein a day up to 5,000 milligrams daily, so a huge range there. And they primarily reported successful desensitization using different criteria for desensitization, and the success rate ranged from 21% to 49%. But again, no standard definition there. And then there was a systematic review and meta-analysis recently published in Lancet, at least electronically published, and I'm going to summarize the results of that meta-analysis of oral immunotherapy. They had 12 randomized trials that met their inclusion criteria. The trials randomized about a little over 1,000 patients, and the median age was 8.7 years. To put in context, the median age in the Therapies trial, I believe, was 7. In the phase 2 trials of AR101, the median age was also about 7. So this is representative, roughly, of the patient population studied for the therapies we're considering. And in this analysis, there was net harm during the trials. There was a 3-fold increase in the odds of anaphylaxis in the trials. And it didn't matter whether they were looking in the buildup phase, so that dose escalation phase, where you might expect more reactions as the dose is increased. So no difference between that dose escalation phase and the maintenance phase. So we would hope that over time in the maintenance phase, the number of reactions would go down. That would be the goal of desensitization. Epinephrine use was also increased, about a 2-fold increase in the odds of epinephrine use, and serious adverse events were also more common in the treatment groups. And unfortunately, probably the most important benefit of these therapies, because we're not talking about a mortality outcome or an increase in life years, the most important benefit would be improvements in quality of life for the children who are affected and their caregivers. Most of the trials didn't report any outcomes on this. 4 did, and there were no significant differences between those in the placebo group and those receiving desensitization therapy with oral immunotherapy. So the conclusion of this trial was that in patients with peanut allergy, there was high certainty evidence showing that available peanut oral immunotherapy regimens considerably increase allergic and anaphylactic reactions over avoidance or placebo, despite effectively inducing desensitization. So that sort of sets a context for us where there's some concern about desensitization as a surrogate outcome for what we really care about, reducing reactions to accidental exposures to peanuts in patients treated with these therapies, and improvements in quality of life for both the patients and their caregivers. So with that context, I'll turn to the phase 3 trials. I'm really focusing on the phase 3 trials because they use different inclusion criteria and had different outcomes than the phase 2 trials. For AR101, the phase 3 trial is the Palisade trial. I already noted that they enrolled patients between the ages of 4 and 17, and fully a third of the patients were in that 12 to 17 age range. And this is important because there's consistent data in the literature showing that the younger you're treated, the more likely you are to become desensitized. So these therapies appear to be more effective in younger individuals. So there are more older individuals in the AR101. And in terms of inclusion criteria, the eliciting dose in the Palisade trial had to be less than or equal to 100 milligrams of peanut protein. Viaskin, the peanut, the phase 3 trial is the Pepetes trial. They enrolled slightly younger children, ages 4 to 11. The median was 7 years, and the eliciting dose went up to 300 milligrams in the Viaskin Peanut phase 3 trial. The primary benefit, again, is desensitization. The definition of desensitization was different in the two trials. In the Palisade trial, patients went through a double-blind oral food challenge, so they actually also were given placebo and went through this as well to really see whether it was the peanut protein that was eliciting the reaction. And the definition of desensitization was tolerating 600 milligrams of peanut protein. In the Pepetes trial, the outcome varied depending on the sensitivity of children to peanut protein at enrollment in the trial. So if they had an eliciting dose that was less than or equal to 10 milligrams, they had to have an eliciting dose of at least 300 milligrams to be considered desensitized. And if their eliciting dose was greater than or equal to 30 milligrams, then to be considered a success, to be considered desensitized by the therapy, they had to have an eliciting dose, of 1,000 milligrams of peanut protein in the oral food challenge. And the success in the two trials, the difference was greater in this primary outcome for AR101, 67% in the active treatment group, 4% in the placebo group, whereas for Viaskin Peanut, it was 35% versus about 14%. And I think the placebo response rate is important here. The patients in the Pepetes trial tolerated on average a little bit more peanut protein and had to reach a slightly lower threshold. So it's not surprising that a higher proportion of those patients actually met the definition of desensitization even though they got placebo therapy. That was less true in the Palisade trial where they were more sensitive to begin with and had a higher bar on average to be considered desensitized. And unfortunately for both trials, there are no quality of life outcomes reported in the trials, particularly for the placebo comparison. And a final little subtle point, even though both comparisons for the primary outcome, the p-value was less than 0.001, so highly statistically significant, in the agreement with the FDA, the primary outcome for the Pepetes trial was that the lower bound of the confidence interval for the difference from placebo had to be greater than or equal to 15%. And in this case, the confidence interval for the difference from placebo ranged from 12.4% to 29.8%. So they didn't meet their pre-specified primary outcome. Whether that's important in our deliberations, we can talk about, but I wanted to point that out because it was an important consideration for some people in thinking about the outcomes here. Primary harms, as in oral immunotherapy, severe and serious AEs were more common in the active treatment group for both the Palisade and Pepetes trial, so for both AR1 and Viaskin Peanut. Discontinuation rates were higher with both therapies, 21% versus 7% for AR101 versus placebo, 10% versus 9% for Viaskin Peanut versus placebo. And the discontinuation rates clearly due to AEs were reported to be much higher for AR101, so almost 12% versus only about 2% for Viaskin Peanut. Epinephrine use also was more common in the treatment groups in both trials, so 14% versus 6.5% for the placebo group in the comparison in the AR101 trial, 9.2% versus 3.44% in the Viaskin Peanut trial. Controversies and uncertainties, I think, the primary benefit of these therapies should be quality of life, but there are no results reported in the trials to date. We have a surrogate outcome, desensitization, for which there's no standard accepted definition, and we saw 2 different definitions used in the pivotal trials for the 2 therapies. And even within manufacturers, so the different Viaskin Peanut trials use different outcomes, and the same thing for AR101. More importantly, there are uncertainties about long-term outcomes. We hope that desensitization would translate into lower rates of systemic reactions due to accidental exposures over time and lower rates of use of epinephrine and so on. But we don't have data supporting that at this point. Long term, we would hope that these therapies may lead to sustained unresponsiveness. That's ability to tolerate exposure to peanut protein when no longer on the therapy. Currently, the thinking is that we need to continue these therapies for years. For patients, it's not clear at this point whether we're ever able to withdraw the therapies, and certainly in the shorter term trials when these therapies are stopped, within a few weeks, most patients start to become responsive again to peanut protein. And finally, tolerance, which would mean really completely able to tolerate peanut protein. That would be the ideal, and it's not clear that these therapies will lead to tolerance at all. So in summary, for AR101, there was a high rate of desensitization, but many dropouts. 20% of patients dropped out of the trial during the first year. There was an increase in adverse events, especially GI. I didn't go into this in great detail, but oral therapy tend to have more gastrointestinal side effects, and clearly more epinephrine use, and no data on quality of life. So our overall assessment is that this is promising. We hope that desensitization will turn into a positive outcome, but it is possible that the net harms we're seeing now are real, that there is a possibility that these therapies have net harm for patients, but we hope that this will translate into long-term benefits. Because of the possibility of small possibility of net harms, we consider this promising but inconclusive. So the net benefits, the benefits minus the harms, we really are unclear how large those benefits would be. For Viaskin Peanut, there's a lower rate of desensitization by the definition used, so the absolute benefit in terms of desensitization is smaller, but fewer dropouts due to side effects of the therapy, and most of those adverse events were dermatologic related to the patch. Unfortunately, epinephrine use was also significantly higher in the patients treated with Viaskin Peanut versus placebo in the PEPITES trial. And so, again, we have significant uncertainties about long-term benefits, the potential for net harm, at least as seen in the clinical trials to date. So we rated the therapy as promising but inconclusive. I mentioned the new data. So the Artemis trial is an AR101 randomized trial, similar to the Palisade trial, except that we had 9-month outcomes rather than 12-month outcomes, 175 children between the ages of 4 to 17, and we see significant rates of desensitization to 300, 600, or 1,000 milligrams of patient protein. But again, we see an increase in systemic allergic reactions and an increase in epinephrine use in the active treatment compared to placebo. So we still see net harm in the trial. There's also an extension trial, the Palisade study, that reported on AEs, and in the 37% who continued on therapy and were reported at this meeting in Portugal, you had similar rates of AEs out to 18 months as were seen during the clinical trial. And there was a report on quality of life just in patients who were initially on AR101 and continued for 6 additional months. This is only a third of the total number of patients who were randomized to AR101. And we don't have comparisons with the placebo group. All we have is comparison with placebo, and they did experience a quality of life gain, but we have no comparisons to what happened to patients who were in the placebo group. So in summary, AR101, higher rates, did we already do this? How do we do this? I think I've already gone through this for AR101, promising but inconclusive, Viaskin Peanut, promising but inconclusive. Oral immunotherapy, we felt like there was insufficient evidence in at least the trials that met our inclusion criteria to really say anything conclusive. They were poor quality trials, open label, and a comparison among therapies, no head-to-head trials, different patient populations, different outcomes, so we didn't feel like there was sufficient evidence to make any firm conclusions in comparing the different therapies. We received a number of public comments. Some felt like AR101 should have a higher certainty rating. This was mostly, I think, focused on the fact that there was high certainty that desensitization rates were high with AR101. But our rating of the certainty evidence is around the net health benefits. So when you fold in the harms with extra epinephrine use, adverse, increased systemic allergic reactions, and so on, we felt like there was a lot of uncertainty, and so did not give it a high certainty rating. There was some concern about our report about accidental exposures, that it was based on old data. In the references, it's a trial of almost 800 American children. It's a cohort study. It followed for 5 years, so we actually have incidence data, and it was published in 2012. There were comments that we underestimated the risk of dying in adolescents. We did highlight that adolescents are risk-taking, and most of the deaths due to peanut exposure do occur during the teen years, so we tried to highlight that. But overall, the mortality from peanut allergy is low in the United States. And finally, we received a number of comments that we did not highlight in the report, that both AR101 and Viaskin Peanut received both fast track and breakthrough designation by the FDA as far back as 2012, so we folded that into the report, and I just wanted to highlight that here. So with that, I think I can take a few questions, Steve, yeah, or open it up for other discussion.
I'll ask, as we get going, as we get multiple questions, please maybe turn your name label up, and we'll go in that order.
Sorry, just wanted to go back for a moment to the quality of life data, which seems like it's really sparse, but there was a little bit from the follow-up of the Palisades trial. Did I get that right? You were going pretty fast. So.
Yeah, sorry. So yeah, for the Palisade trial, which is the phase 3 trial for AR101, we don't have any reports published or presented yet comparing the active treatment group to the placebo group, but there was a long-term, an extension trial, and about a third of the patients originally randomized to AR101 continued on AR101, and we have data 6 months after the end of the trial stating that they had significant improvements in quality of life on a standard food allergy questionnaire, quality of life questionnaire.
Compared to baseline.
Compared to baseline. So not compared to the control group, it's compared to baseline. So we don't know whether, the patients in the placebo group who continued therapy and did well, what their change in quality of life would have been over the same 18 months.
And can you give a sense of the absolute value of that improvement?
So I don't recall. I mean, it was large enough to be considered clinically significant. I don't recall the exact numbers. Maybe some of the experts know the difference in the FAQ, LQ, and.
So just to clarify with quality of life, so the difficulty is when you have both groups concealed and blinded to treatment assignment, it's almost an exercise in futility to try to measure quality of life in that first year. So really, while in a perfect world, they would have quality of life data at year 1. Essentially, everybody thinks they're on treatment, so it's kind of a futile exercise. So really, they need to let it go a year or two at that point. So that's why it's coming out delayed. And while one group has published an abstract, again, not peer-reviewed, not submitted for publication or anything like that, and the other groups is forthcoming, you're looking at quality of life being measured in two types of indices. The first one, so this is the FAQ-LQ. So this is a European index that has some validation in U.S. populations, but was not designed for use in U.S. children or by U.S. parents rating their children. So you have a parent proxy index rating the quality of life of kids aged 0 to 12. and then you have a child index rating in the child rating their own quality of life from 8 to, is it 12 or 16 or something like that. The data that was presented in Lisbon showed that the child form, so the child's rating, that was the one that was significant and not necessarily the parent's rating. I was trying to pull up the abstract. And then DBV's data is being calculated and is forthcoming. And again, they'll have their own side of the story. The issue with quality of life is whose quality of life matters most. Is it the child's report? Is it the parent's impression of what the child is going through? Or is it the caregiver burden? In the US, the validated index that was developed for use is really the FAQL-PB, the Food Allergy Quality of Life Parental Burden, developed in 2004 at Mount Sinai. And that's measuring the parental burden. So these are the drivers of the healthcare services in these kids. They're the ones making the decision that I want therapy. It's rarely the child saying, hey, how about I go on this, or I go get this vaccine today, or whatnot. I'm sure there's somebody out there who might say that, but probably not. So again, we have to just be careful. They are the available indices out there. Quality of life might not actually even be the right metric that we're looking at. Again, it's not a health utility, so there's no sort of assumption of risk or preference under a condition of risk. And it also doesn't necessarily deal with their self-management capabilities, which might be the thing that shifts the most. So something like self-efficacy might be a better tool. But the absolute mean clinical difference, so that was, of course, validated. It was Gordon Guyot's group at McMaster's, right? So they said that any 0.5 difference on a 7-point index is called a minimally clinical important difference. That's actually never been tested to show that it holds across different disease states, including food allergy. When you look at other measures, like a standard error of measurement or something like that, then the minimal difference is probably closer to 0.3, but none of this has really been validated in this. So it's hard to say what's an important difference, and I don't have the absolute mean numbers of what the difference is. Somebody from AMU might have. Steve, do you have that?
Well, it was greater than 0.5, and I don't have, it was across every domain as well. That's what I.
Well, thank you. I think we'd like to get to a few other questions here. I have a brief one, and that is any background on why the FDA was still going to go with desensitization, as it sounds like their main endpoint. Is that correct? As they've switched for diabetes drugs and other things, realizing they really want to try to get at a more clinically relevant endpoint.
Yeah, I mean, I can't speak for the FDA, so I don't really know their thinking, but I think given, breakthrough designation and fast track, they allow, a surrogate measure, but then hopefully, and we don't know in food allergy, they would require a sort of a clinical outcomes piece as, the sine qua non for continued approval. As you know, for lipids, the lipid lowering, you get approved, but you have to do a long-term trial that shows a reduction in strokes and heart attacks, which is really what matters to patients, and you would hope that that would be the same here. I don't know whether the FDA will require that in this case.
Thank you. I think we'll go Elizabeth, then Ralph, and Rena.
Thanks for that review, Jeff. So a couple questions, but first, just quickly for the quality of life, surely they have the data for the control group and the other groups. So I guess it's more of a comment, but I find it very curious that they only represent a small subset of what must be a much more complete data set.
Yeah, I mean, I think, again, as Dr. Greenhot pointed to, I think at 1 year in a placebo-controlled trial, is unlikely to be a big difference.
Right. So, and to just make sure I understand things, we have a treatment that doubles or in some cases triples epinephrine use and anaphylactic reactions, which in theory is a treatment for that same thing. The disease has a huge impact and huge burden for families, right? And so if they're, even with that, if there is relief there, that would be hugely helpful, but we have no data on that. I just want to make sure I understand that correctly.
I think that's correct. To date, we don't have really clear data that desensitization by any metric of desensitization translates into the clinical benefits that we care about, that patients and their caregivers care about.
Okay.
Ralph?
I'd like to touch on the area of medical adherence. So, Jeff, you talked a little bit about the increased burden related to adolescents, risk-taking, leaving home, this sort of thing. And you also mentioned the issue that you lose the desensitization process after a couple of weeks. So I'm interested in, these were randomized clinical trials. In the real world, we'll assume that there'll be medication adherence issues. Well, I would be interested in the expert panelists, whether that would further increase the potential chances of harm related to the medications versus potential benefit, and and we won't even talk about cost because we don't have the issues of cost, because that would involve repeated formal desensitization with AR-101. But perhaps learning more about your perceptions of the issue of medication adherence in this disease would be helpful.
Sure. So it goes without saying that drugs don't work if you don't take them. Obviously we want people to adhere to their treatment, but like very few people actually take the medicines as directed. The issue here is what happens with, desensitization loss or potential sustained unresponsive loss. And we know it is a ticking time bomb. Like, take the case of a penicillin allergic patient that we desensitize. They have a window of about 36 hours after we get them up to a point to continue to see that drug or lose the benefit of the desensitization. Now, this has not necessarily been well tested within food, but it's probably following along similar therapy lines, that you need this constant exposure. So the more that they miss it, the more susceptible, some of them at least, I don't know if it's all, and there might be some who hold on to it longer or whatnot, and just like everything, there's heterogeneity of treatment effect. But yes, if they're not taking this, and this is something that's designed to be either applied or taken orally every day, then yes, they do risk more harm. But we haven't really figured out who's able to stretch it out and who's going to be okay. I know that from allergen immunotherapy, when we give like ragweed or venom or whatnot, you're on a very strict schedule, and we do see more adverse events when patients don't come in every week. Now, it's a different timetable, but that signal, you can stretch them out to a certain point, but again, there's a very narrow threshold that they work within. But we don't have the exact evidence to answer your actually very, very good question here.
Rena? Sorry, you want to?
I just wanted to chime in here a little bit on the patient quality of life issue. I don't think it's debatable that we need more data and that we need better tools as well. So I think that's part of the conversation. It was in the evidence report, and it's part of what we just discussed now. And I think that's absolutely right. And what I hope to bring to the conversation today from the patient perspective is, in absence of perfect data, what can we learn from the experience of what desensitization means to a patient, and why is that valuable? And the question came up, why would the FDA designate this a breakthrough therapy with fast-track status? I think our interpretation of that would be desensitization is the end goal that patients are looking for, because when you desensitize, you relieve that daily frustration and chronic burden that our community lives with. And that benefit of quality of life accrues greater over time based on the experience of patients who have undergone immunotherapy. So we do need more time to measure that more accurately. We need better tools. And in absence of having that perfect data to make these types of assessments and judgments on policy, I would encourage everyone to really hear the patients and what do they say around with the benefit? How did it change their life? Why was it worth going through that risk for increased reactions to get the benefit at the end of that protocol? And I would really encourage us to think about that and to fill the missing information gaps with testimonials and as much input we can gather from the community. Thanks.
Thank you. We have time for one more question. And for those of you who've been patiently waiting, we will have an opportunity later, either in the morning or after lunch, for you to ask your question.
I'm just wondering are there other examples of food allergies with anaphylactic reactions where the surrogate outcome of desensitization was later shown to have improved quality of life in trials?
No, not really. Yeah, pull up the data. All right, he's pulling it up, but not that we know of, and he's going to pull up some data on the other foods. But I do want to just really quick clarify. We've both written a number of papers on quality of life, and I just want to point out, when you think of conditions, I think food allergy is really unique in the sense that these kids, you don't see their disability when you look at them, but think about food. Everywhere they go, there is fear. And so what we've published is the intensity of that quality of life and that anxiety and that fear in not only the parents, but in the children, especially as they grow up, and especially if they've had an anaphylactic episode. So I think, just measuring this out and comparing it to other conditions is very difficult. I'm a generalist, so I see everything. And from food allergy, it is by far, and we've published this, one of the hardest conditions for kids to live with because it's in everything they do, and you can't protect them all the time, and you need their world to help protect them from an accidental ingestion. I also wanted to clarify a couple points from the beginning. From our data, 40,000 kids across the United States, we found that peanut allergy, only about half of these kids with peanut allergy have multiple allergies. I know that came up. The other thing I wanted to point out is that peanut allergy was the highest one that reported severe allergic reactions. So of all foods, kids with peanut allergy had reported having a severe allergic reaction. Also, 41% of the kids with peanut allergy had reported using epinephrine. And I don't know why we think of epinephrine as so bad. It's a very safe medication. It's not like we're injecting them with steroids or anything really dangerous. Steroids are, they're worse than epinephrine. That's the point. We do that all the time. So what thinking of epinephrine as this, horrible adverse event is not necessarily what we should be considering. And I also want to, just because access is so important to this group, I do want to talk about the fact that 2 times as many peanut allergic kids come from households earning less than $100,000. So, and it's significantly greater in non-white kids, twice as many. So just taking into context, because I know that wasn't discussed at all, this is a condition that does impact underserved minority and low-income children substantially, and those are the kids that are not. They're also the kids that are not seen, that are seen more in the emergency room and are not getting proper care and are not able to afford all the special diets and all the safety precautions.
So the data from other allergens is limited. Like most of the OIT studies and most of the studies for therapy have been focused on peanut. Milk and egg sort of launched OIT studies, and then most of that research has tapered off since about 2012 in the U.S. So what you have are the private groups doing a clinical service and not researching it in terms of efficacy. Outside the U.S., the group in Tel Aviv has shown that there are similar effects for other allergens, but most of the data has clustered to peanut because that's the one that's, honestly, of all the OITs, it's worked the best and it's had the most interest, so that's why you're seeing it. The one thing I would also caution about with quality of life is that the net benefit of how much somebody is desensitized is probably not linearly tethered to sort of how much their quality of life is going to improve. We did a qualitative study of our trial participants from both studies, and what they showed is that they honestly didn't care how much they were desensitized. They just wanted an upward movement. So even small gains were seen as providing meaningful benefit to them.
So, thank you. And thank you for all of your comments. And Jeff, we'll have you introduce our next speaker.
Right, yeah, no, I just think it's perfect. Our next speaker really is going to give the patient and caregiver perspective. Kenny Mendez from the Asthma and Allergy Foundation of America, and he's going to present results that hopefully will give some grounding of that from a survey that they did in concert with us for this meeting. Thanks, Kenny.
Great, thanks, Jeff. So, as Jeff just said, I'm Kenny Mendez, the President and CEO of the Asthma and Allergy Foundation of America. We're the oldest and largest patient group. We've been around for 65 years. We try and elevate the voice of patients, and both on the asthma side, and I feel like my conflict of interest here is I feel like I work for ICER since I've been through the asthma study with Jeff and others earlier on. But we did a quick qualitative study of 3,000 of our community, 800 responses. And I'm going to go through this really quickly. You have the data in your decks, but in the interest of time, just want to get through this. Clearly, there's no quality of life impact in some of those things, but what we try and do is call out what some of the burdens are from our community, and clearly caregiver burden is one of them. You'll see fear, anxiety, those are the words that come up here. Clearly a financial impact, there's a financial burden for those. We talked about the underserved, but we'll get into the specific numbers. I've got some slides later on. And whether or not peanut desensitization makes a difference? Yes, it does, and we'll get into that in a second here. So here are the numbers behind some of this. These are the four areas that we kind of surveyed: mental health, social life, emotional health, and travel and vacation. So does desensitization, does food allergies have have an impact? And you'll see that it does. From 3 on to the left, it shows that there is some sort of impact there. This slide was pretty interesting to us in terms of the caregiver anxiety. It causes fear and anxiety in my child, 64%, but then it causes fear and anxiety for me and my family, which is close to 81%. So that really shows you and demonstrates the caregiver burden that we've been all talking about here. Again, the words that come up are fear, worry, and unease. You'll see down the left-hand side some of the questions that we asked, but clearly over 54% is always and often, and then occasionally. So it's very much on your mind if you have food allergies in the family. Over 25% here have seen, mental health professional to deal with food allergies in the family. And then in terms of how often do you think about it, 82%, it's on your mind all the time if you have food allergies. In terms of the financial burden, here are some, questions that we asked: specialty foods, cost of out-of-pocket epinephrine, and the cost of food challenges. But in all cases, significant impact or major impact on having food allergies and the financial burden. You'll see that 41% said it's a negative financial burden. And as we hear from patient advocates later on, you'll hear about how some of us have made life decisions based on our kids' food allergies. In terms of a negative financial impact on your household, 81% have said that it has had a negative financial impact. And then if you look at that, these are the various areas, but the one that jumps out the most is my family incurred more credit card debt because of food allergies. As you look at time burden as well, shopping for safe foods, reading labels when you're in the store, understanding which restaurants are safe or not, that represents significant time burden and effort on behalf of those who have food allergies. You'll see here in terms of the last 12 months we surveyed missed school days, over 55% have at least missed one day of school or more because of food allergies. So the bottom line here is desensitization is something that's very much important to the community and would have a positive impact on quality of life. While we were talking about it here earlier, our survey among our community basically is borne this out. These are pretty detailed questions. This one is one that we worked with ICER to put into some of the survey, which affected some of the utility measures in their analysis. But this one really asks about desensitization, does it have an impact on your quality of life? And 92% said that it would have some impact on quality of life. And then this one was intolerance to peanuts specifically, and you have here 97% said they would have an impact on their quality of life. So in terms of the societal impact, which I think is what's really important from this exercise, because previously we heard about the model and all the uncertainties in the model or the calculations in the model. I mean, I think the most important thing from this exercise, and I think we're talking about it now, is the societal impact of food allergies, and that we really do need more information in the future, but accidental exposures and the psychosocial aspects of food allergy are fairly significant for caregivers. The survey results unmet needs for caregivers. You know, we talked about mental health, all the extra time spent in terms of caring for your family. And then in terms of the larger economic impact on society, those studies aren't done yet, but clearly there are larger economic impacts. And again, I think we'll hear about some of that from some of our patient advocates. And then in terms of quality of life, I think we mentioned this, it's a silent disease. A lot of people don't know about it, but I think it's very important to amplify that voice and really understand the impact of food allergies in general, both from a financial, social, psychological point of view. So I'll pause there for any questions.
I think this will be maybe I'll say let's hold the questions till after we hear the economic model, because I think a lot of this ties into the modeling. And at this point, I think let's go ahead with, thank you, we'll have Dr. Greg Viaskin from the University of Washington present on the economic model.
Okay, hello. I'm Greg Viaskin from the University of Washington in the Comparative Health Outcomes Policy and Economics Institute. My colleague, Dr. Ryan Hansen, and I did the cost-effectiveness modeling for this project. We do receive some financial support from ICER to do these economic models for them, but otherwise the two of us don't have any industry funding related to peanut allergy that we need to report today. Our objective was to estimate the lifetime cost effectiveness of 2 peanut allergy immunotherapies using a decision analytic model. I'll just go over some methods quickly just to orient everyone to what we did. Our overall approach, we compared AR101 from Immune Therapeutics and Viaskin Peanut from DBV Technologies. These were both compared to avoidance alone strategies. The base case standard of care was peanut avoidance alone. The patients in each comparator are assumed to require lifetime access to epinephrine unless they outgrow their peanut allergy. And we compared the immunotherapies to avoidance alone only, but not to each other, for the reasons that Dr. Tice outlined as far as differences in patient populations and outcomes. Our modeled population was children age 7 with a peanut allergy. We developed a de novo Markov model, which I'll explain on the next slide. Our setting was United States, a healthcare sector perspective. We looked at a lifetime time horizon, a discount rate of 3% per year for cost and outcomes, and that's just saying that costs and outcomes in the future are valued less than costs and outcomes that are more close to the present. Cycle length of, we modeled weekly for the first year, that's the trial duration for both trials, and we looked at weekly cycles so that we could get a more granular look at the costs and outcomes from the trial. And then after that first year, we then switched to annual cycles to model the lifetime outcomes. The primary outcomes were lifetime costs, quality-adjusted life years gained, and incremental cost-effectiveness ratios, which are the cost per quality-adjusted life year gained. However, and those are in the report, but for this presentation, we'll be focusing more on the value-based prices for these, since we don't have the actual costs yet for either one of these therapies. I'll also note that typically we would present the equal value of life year gained for our comparators here. But we did not model any difference in mortality among all of the comparators in the model. So mortality, or survival, if you will, is the same for both the immunotherapies and for the avoidance alone strategy. And so we didn't calculate the equal value of life years gained. So here's our model. It's a Markov model. I'll walk us through it. You can see that we have 5 different health states. Patients begin the model in the top left corner for treatment plus avoidance, or avoidance alone with peanut sensitivity. Within that first year, and I mentioned already that those were weekly cycles, patients can either remain in the health state in which they are. So if they're in the treatment group, then they are being treated. The cost of the treatment is applied. The adverse events associated with treatment are counted. There is a slight risk for all patients for an anaphylactic reaction that leads to death. So that's a possibility, and that's that bottom arrow. And then the third arrow there is just representing trial discontinuation. And so we're modeling patients who are on treatment and then eventually moving off treatment during that first year. At the end of year 1, there's the exit food challenge, where the patients are tested to see whether or not they can tolerate the dose of peanut protein that they're given. If they successfully pass the exit food challenge, then they move into the peanut desensitized health state. If they don't pass the food challenge, they move into the untreated with peanut sensitivity health state. And then for the remainder of the model, we're using the annual cycles again, and patients can remain in the last two treatment states that I described, the untreated with peanut sensitivity or the peanut desensitized health state. And we're assuming that if you are in the peanut desensitized health state and you're on immunotherapy, then you remain on immunotherapy for the rest of your life. There's still the risk of death from anaphylaxis, but also just background mortality over time, since this is a lifetime model and we're modeling patients from the age of 7 all the way until theoretically the age of 100. And then there's the possibility from both of the alive health states that these patients could eventually outgrow their peanut allergy, at which point they would enter the peanut tolerant health state. And in that health state, they've got the highest possible utility values that we're using in the model, and they go off treatment. Some of our key model assumptions. We achieved that desensitization leads to better quality of life. And this is really the biggest assumption in the model, since we don't have, as has been described, we don't have the data on utilities as yet. We assume that individuals who successfully complete AR101 or Viaskin Peanut trial regimens and pass the 1-year exit food challenge remain on maintenance treatment indefinitely unless they outgrow their allergy. Individuals who pass the 1-year exit food challenge are considered desensitized regardless of the treatment arm, and that's because some patients in the placebo arms in the trial ended up passing the exit food challenge even though they weren't on immunotherapy. So we just assumed that if that happened, they were considered desensitized for life. And lastly, we assumed that individual behaviors such as increased risk-taking, once a patient is considered desensitized, we assume that doesn't change based on whatever health state that they're in. These are the clinical inputs that Dr. Tice has already gone over. As you can see from the Palisade trial, 4% of placebo patients passed the exit food challenge. And so that's 4% who entered the desensitization health state in the avoidance alone group. AR101 with the 63.2% improvement over that. And so in the model, we're just adding the 4% plus the 63.2% to get the total number of AR101 patients who are desensitized. At the bottom in Pepetes, is 13.6 for placebo, and then we're adding the 21.7% for Viaskin Peanut. As I've mentioned, we don't have costs for these drugs yet. They have not come onto the market. They're not approved by the FDA yet. And so what we had to do was figure out what would be the best estimate that we could find. We ended up finding some analyst estimates online that we used in the model, and these placeholder costs are in the report, but I will not be discussing any of the cost or incremental cost effectiveness ratio outcomes based on these today, just because they are placeholder. We needed a cost for the model in the report, and these are the ones that we used. This is treatment discontinuation and how it was modeled in the trial. As you can see in Palisade, with AR101 getting the higher dose of peanut, you can see that they have a higher discontinuation rate versus placebo or avoidance alone in our model. Below that, Viaskin Peanut with the much lower dose of peanut tracked a lot more closely to the placebo or avoidance alone strategy. There were just the discontinuation rate was lower. For the adverse events, we applied cost based on the CMS final rule table for 2019. Those ranged from about $3,500 to $9,300 for anaphylactic reaction. We also applied a disutility of 0.311 for 3 days per event. And then I'll just have a note at the bottom that we did model a pretty significant reduction in anaphylaxis risk of 95% if the patient is desensitized, but 95% of an already sort of low probability for a major anaphylactic reaction didn't lead to much reaction being prevented. These are health state utilities. Again, it's the same model. So what we had to do was find the best estimate that's out there given the lack of preference-weighted utility studies in the food allergy literature. Not only peanut allergy, but as it was mentioned by the clinical experts, there just aren't any preference-weighted utilities for food allergy in general. We found a source, Swedish source, food allergy. It's a convenient sample comparing patients without food allergy to patients with food allergy. And we got these utilities, the estimates were stratified by age, so we've got different utility scores for age 0 to 11 and age 12 plus. And so if you are peanut sensitive in our model for the two health states here, in your age 0 to 11, your health state utility is 0.84. If it's age 12 plus, your health state utility is 0.91. For the peanut tolerant health state, we had an estimate from the same study, and this is for patients who don't have any food allergy, which makes the most sense. If you're peanut tolerant, we're assuming that you're cured in the model. And so we applied the utilities for no food allergy from the same study. So that's 0.94 for age 0 to 11 and 1.0 for age 12 plus. And then the question was, what do we do for the peanut desensitized health state? And this is really, the estimates that a lot of the model results kind of hinge around. And, we heard a lot of opinions from multiple groups. Initially, we started with just saying it would be a 50% improvement from peanut sensitivity, between peanut sensitivity and peanut tolerant. And that was perhaps a bit arbitrary, and we heard from some clinical experts that that the utility value should be closer to sensitive. We heard from some other groups saying it should be closer to peanut tolerant. But based on the work of the Asthma and Allergy Foundation, we came up with this estimate that of a 60% improvement based on a survey that they conducted during the course of our process of building the model. And so we ended up with utilities of 0.90 for age 0 to 11 and 0.964 for age 12 plus. We included a societal analysis. This was not our base case analysis, but it was done in addition to the base case analysis. We included annual out-of-pocket costs and annual productivity loss for a total annual cost of $3,330 per patient. And then we had the same problem finding utilities for parents and caregivers in this population, that wasn't available for patients. So clearly it would not be available for parents or caregivers too. And so what we did was we used 0.901 for peanut tolerant. The anchor there is the utility of a relatively healthy 30 to 39-year-old person in the United States. And then from there, we subtracted a assumed range of potential disutilities from 0.01 to 0.05 to get us to the treatment or avoidance alone with peanut sensitivity health state. And then from there, to get to the desensitized health state, we applied that same 60% improvement that we used for the patients. So the lifetime outcomes and value-based prices for AR101 versus avoidance alone. AR101 led to 27.19 quality-adjusted life years. Avoidance alone, 26.44 life years for an incremental difference of 0.75 quality-adjusted life years. As I mentioned earlier in the presentation, we didn't model a difference in survival among any of the treatment strategies, so there's no difference there. We came up with a value-based price of $50,000 per QALY for AR101 of $2,369. If the threshold for cost effectiveness is $100,000 per QALY, the value-based price was $4,808. And at $150,000 per QALY, the value-based price was $7,248. For Viaskin Peanut versus avoidance alone, quality-adjusted life years were 26.81 for Viaskin Peanut, 26.55 for avoidance alone, and that gave us an incremental difference of 0.26 quality-adjusted life years. Same survival or life years as the previous slide. The value-based price for Viaskin Peanut at $50,000 per QALY was $1,508. The value-based price at $100,000 per QALY was $3,010. And then at $150,000 per QALY, the price was $4,513. In sensitivity analyses, this is one-way sensitivity analysis where we vary each parameter by a range from a low to a high to see which parameters have the biggest impact on our model results. And clearly, in both analyses, the biggest drivers of our results were the utility values that we used in the model and the costs of the treatment. The efficacy of the treatments did play a role. As you can see, they're listed here in both analyses, but they were not as important as our utility values and the cost of the drug, whatever that might end up being. We performed a number of scenario analyses, and we found that the incremental cost effectiveness ratios were generally stable, meaning there were no meaningful changes as far as whether or not these were cost effective when we extended the treatment discontinuation rates beyond the trial duration. Patients were discontinuing and moving. Basically, we removed the cost of treatment, but we didn't have any long-term data on whether or not they then reverted back to peanut sensitive. And so essentially the cost is taken away, but we didn't make any assumptions there in that scenario analysis about effectiveness. We turned off the probability of spontaneous tolerance, but that equally impacted all four of the modeled comparators, so that didn't lead to any difference. We varied the risk of accidental peanut exposure, and again, the rate of anaphylaxis and the consequences of peanut exposure are rare, and especially if you're treated, we've got the difference of 95%. The impacts were minimal on the results. We varied the patient age at model entry, since the populations in the two trials were slightly different as far as their inclusion criteria. We employed Palisade secondary outcome because they also looked at, instead of the base case 600 milligrams, they looked at 300 milligrams and 1,000 milligrams at the exit food challenge. And then we also varied epinephrine utilization by plus or minus 50% in the years 2+. And for each one of these that we didn't notice any major changes in our results. For the modified societal perspective analysis, we did see some changes in our results when we just added the societal costs. And so this is before we've added any utilities to the analysis. This reduced the incremental costs because you've got more patients in the immunotherapy arms who are desensitized. It reduced the incremental cost effectiveness ratios since we're reducing the cost. And then this led to increased value-based prices per year. AR101 ranged from $5,300 to $10,200, and Viaskin Peanut ranged from $3,300 to $6,400. Then when we added in the parent caregiver utilities to the analysis, so keeping the cost above built in, this increased the incremental QALYs for immunotherapies. It further reduced the incremental cost-effectiveness ratios, and then it further increased the value-based price per year. As for AR101, that went up to $6,100 to $12,600. For Viaskin Peanut, that went up to $3,800 to $7,800. And that ranges from $50,000 willingness to pay per QALY to $150,000 willingness to pay per QALY. We had a number of limitations. The utility estimates used for the base case model come from a food allergy, but not necessarily a peanut allergy patient population. Again, there just aren't any preference-weighted utility values for peanut allergy. Our assumption of a 60% utility improvement for desensitization was perhaps a bit optimistic. We certainly heard both sides of the story, but ultimately the best evidence that we had was the survey conducted by the Asthma and Allergy Foundation, and so that's what we settled on for that. We lacked any long-term treatment data to inform decisions such as the duration of treatment, hence our decision to assume that patients continue treatment until they die as long as they're in a treated health state. And we had no data to tie desensitization to patient outcomes, such as a lifetime reduction in reactions due to accidental exposure. We received a number of comments. Obviously, we got a lot of feedback on our utilities and our assumption for desensitization, but we went with the best estimates that we were able to get a hold of. We got some feedback that immunotherapy placeholder costs in the report should be the same because we did model it. They were a little different based on the analyst estimates. That's not entirely important here because we're not making any decisions based on value today, but I just wanted to put that out there. And then we did make our model available to the manufacturers. It's a relatively new program that a lot of the programs working with ICER are doing, where we give access to the manufacturers so that they can take a look and find if we made any errors, if there are any questions that they have about our approach. And we did receive some clarification from one of the manufacturers about AR101 was the patients were going to too many clinical visits during the ramp-up period of the dosing, and so we were glad to get that feedback and get that change made in the model. In summary, immunotherapies are more costly, more effective, but unknown cost effectiveness because we don't know the cost, and we're not totally sure about the utilities yet either. Immunotherapies for peanut allergy appear to provide benefit to patients and parents and caregivers versus avoidance, but this is all going to depend on the cost and what the literature tells us that the utility values are. And interesting enough, those are our results were most sensitive to those two things: the utility values and the cost of the immunotherapies. So that's it.
Well, thank you. I think we have time for maybe two questions. I see Alex's name, and so I know you had a prior question.
I'm going to sit over with my colleague and answer.
Okay.
Can we have Ken and Greg go to the front table to answer questions together, please? Yeah, why don't you guys both go up there?
So I think we'll do Alex and Kim.
And, yeah, please.
And that not sure we'll be able to get to other questions, but Alex?
Great. Thank you, Greg. And two questions. First question is, did you model in the tornado plot that desensitization could potentially produce negative utilities? And then, in other words, I'm not quite sure how you found the upper and lower bounds of the parameters on that estimate, and whether you went potentially low enough. And then I guess I'll just stick with one question so we have time for others.
Sure, yeah, that was one of the first things that, one of the first issues that we had to tackle with our utility estimates, because if you've got overlapping utilities where you've got a supposedly better health state its range overlaps with a supposedly worse health state. And if you're doing a sensitivity analysis and you're drawing a value that is lower than, drawing a value for a healthier health state that's lower than the value for the less healthy health state, your results don't make a lot of sense. And so we, calibrated the ranges of our utility values to make sure that they did not overlap in our sensitivity analysis. And so we settled on approximately 2.5% because that was what gave us the range that didn't give us that overlap.
Thank you. Kim?
Can you just clarify the assumption about 95% risk reduction in anaphylaxis? And then the studies show that they actually had increased episodes of anaphylaxis. So I'm trying to understand why that was so fixed.
Yeah, that came from a risk reduction study that we got from one of the two manufacturers. I'm blanking on which one it was now.
Yeah, it's one of
It's from DBV.
DBV.
It's from DBV.
Yeah.
But this is a modeling exercise that says if you increase the threshold for reacting to peanut allergy, you would, to the degree at least seen in the DBV, in the trial of Viaskin Peanut, you should see up to a 95% reduction in anaphylactics.
Just to clarify
that's
that
not,
further to understand.
That's not accurate, though. Yeah, so what that showed was a risk of reactivity. It didn't imply any severity of the activity. So your question is actually well taken. What is the plausibility for saying that if you hit your eliciting dose of 300 or 1,000, which means that's the dose at which you tip over, and that was the end mark there, or if you hit your endpoint of 600 milligrams with no more than mild symptoms, which could include mild abdominal discomfort, minor vomiting, including a single episode, or a single episode of diarrhea, rhinorrhea, skin, basically kids that still have some symptoms but per protocol were considered tolerant for the endpoint. So how do you derive 95% risk reduction in anaphylaxis? And that's really the hinge here is the health utility here is, in my estimation, is not plausible.
And then the second assumption was that there's no more medical costs after the second year, especially for the AR-101. And yet you'd think that if some kids stop taking it and then need to be like they're not dropping out, they just weren't adherent, so they have to go back for more drug escalation, that would be another cost.
Right. Well, we had just assumed that they do stay on treatment. If you get to 1 year and you are on treatment and you pass the test, then you continue treatment for the rest of your life.
Right.
A great point. Yeah, I mean, we did not model discontinuation and restarting the therapies. That wasn't something we
Can I
included.
sneak in one more? Do we have any idea of the extent to which the anaphylaxis was due to accidental exposure versus the desensitization process? Or what proportion of that was due to that?
I don't think that we did, and we were really in the model agnostic to the cause. We were just using the rates reported.
Right.
Okay, thank you. I think in the interest of time, we're going to move on. We could get one more question. Okay, Steve. Steve gets a question.
I get a prerogative. Partly because we're not going to be having value votes just because we don't have a real price. I just want to stretch this a tiny bit more. A lot of the discussion, though, will continue to hinge on the utility estimate that has been commented on already. So, Ken, I know we gave you a pretty prescribed amount of time. Can you talk a little bit more about the sampling of the patients and families that you went out to for this survey?
We sent out a survey to 3,000 in our community. We do this frequently, something My Life with Asthma as well, to kind of elevate the burden of the disease. And within that 3,000, we narrowed it to about 850 folks, which is the N in some of the slides that you'll see, who are specifically those who have peanut allergies and who would qualify for some of these questions. So 93% of the respondents of the 853 had peanut allergies and other food allergies. And then I think 7% of that group had only peanut allergies alone in terms of how they identified themselves.
Okay. And I'm not trying to, I'm just trying to be scientific about this. The denominator of children, as I understand it, who have a food allergy is about 6 million in the United States.
It's 8% of 74 million. So I'm just, in terms of the sampling, this is a group of patients and families that have been connected to your foundation, maybe.
Yeah,
so they're not, I would say, representative necessarily of the average patient or family.
Yes, you're absolutely right, and that was the challenge, and we appreciate you working with us on this, because I think, as you could tell from the data going into this, there were no studies out there. So I think what ICER did was extended themselves and basically said, okay, if you have a community that you could survey and ask some of these questions, what would that be? And that's what we did, and we went out to do that. So it's not a published study like some of my colleagues up here, but it was an effort to work with ICER in order to identify what some of the burdens were. And I think as you hear from patients in general through this afternoon, this exercise, you'll see, I mean, this makes sense.
No, it was very helpful. And as you said, we had no other good evidence on which to base an estimate of the utilities, but people will continue to talk about it. Just one other quick question. The discontinuation on ARA 101 was assumed to be 0 if you said they passed the food challenge at 1 year. I was looking back at the ARC 004 results, which is a 6-month extension trial of Palisade. At the end of Palisade, 79% of patients were still on the treatment. 6 months later, only 37% were still on treatment, according to the results of ARC 004. Did you know those data when you were modeling the discontinuation? Because it sounds like that's a much steeper discontinuation rate. And I'm just reading off the slide that you presented. That may not have been the full denominator.
There's a misunderstanding. Yeah, we'll clarify that. Randomized.
Right. No, so this was just the percent in this study that continued, and maybe better to have the company comment on it, but it was the percent that continued on therapy in this extension trial, but there may have been others who would have, and maybe the methods can be described in a little more detail. Again, this just came out, and I just looked at the top line results.
Could you please step forward and use the microphone and just address? I was just looking at I think it was 117 out of 314, and just trying to understand what the discontinuation rate was among patients.
I'm Steve Tillis from Medical Affairs and Immune, and the ARC 004 trial was the open label extension. For ethical reasons, there was no placebo arm. The patients that were on placebo were given the opportunity to be treated with active drug. They were randomized into 3 different arms, only one of which was daily therapy. Okay, so that's the data that we have. We don't have analyzed data from the once a week and the twice a week. So we were trying to get at that question of less than daily dosing and what happens. But what we have is the daily dosing data.
All right, thanks. We should probably go ahead then.
Okay. All right. Well, thank you. At this point, I think we'll move on to hear from our manufacturer representatives. So we'll have those two come up to the table.